Have you ever thought about the most successful life forms on our planet? Well, today we're diving deep into the world of the unseen to get to know the tiny rulers of our world, prokaryotic cells. They might be simple, but their design is nothing short of brilliant. Let's find out what makes them tick.
Okay, so here's our game plan. First, we'll get properly introduced to the prokaryotes. Then, we'll look at the protective layers that keep them safe. After that, we're going inside, into the cytoplasm to see the machinery. We'll also check out the external tools they use to move around and stick to things. And finally, we'll wrap it all up with the complete prokaryotic blueprint.
So, first things first, who exactly are the prokaryotes? Well, we're talking about a huge group here. It includes all bacteria, blue-green algae, and these incredibly tiny cells called mycoplasma. And what's wild is that even though they come in all sorts of shapes and have tons of different jobs, they all share the same fundamental organizational plan.
To really get a feel for their world, you have to adjust your sense of scale. Way down. Take a look at this. That big circle, that's a typical eukaryotic cell, like the ones that make up you and me. It's a giant, relatively speaking. Now, see that little guy next to it? That's a bacterium, about 10 times smaller. And it gets even smaller. You've got mycoplasma, which are also called PPLO, that stands for plural pneumonia-like organisms, and then viruses, which are even tinier. Being this small is actually a superpower. It lets them multiply way faster than bigger or more complex cells.
Now, just because they're simple on the inside doesn't mean they're all identical on the outside. Not at all. Bacteria come in 4 basic shapes. You've got the rod-shaped ones, called bacillus, the little spherical ones, known as coccus, the kind of comma-shaped ones, called vibrio, and the cool spiral-shaped ones, which are spirillin.
So, you might be wondering, how does something so tiny survive in a big, harsh world? Well, it all starts with an incredibly tough exterior. We call it the cell envelope, and you can think of it as 3 layers of high-tech armor. This cell envelope is a really tightly bound 3-part structure. If we start from the very outside and work our way in, first you hit the glycocalyx, then you get to the cell wall, and finally, the plasma membrane. Let's peel back each layer one by one.
Okay, first up, the glycocalyx. This is the absolute outermost layer, and honestly, it's a bit of a wild card. Depending on the bacterium, its makeup and its thickness can be totally different, which gives us 2 main varieties.
For some bacteria, the glycocalyx is just this loose, thin covering we call a slime layer. But in others, it's a really thick, tough, and dense layer called a capsule. And that capsule is serious business. It can be the key to helping a bacterium hide from an immune system and cause disease. Right underneath that glycocalyx, we find the cell wall. This is the cell's real body armor. It's rigid, it's strong, and it's what gives the bacterium its specific shape. Most importantly, it provides the structural support to keep the cell from bursting under pressure or collapsing. It is absolutely vital. Except for mycoplasma. They're the rebels of the prokaryotic world and somehow manage to get by without one. Now, this whole cell envelope isn't just for protection. It turns out its structure gives scientists a fantastic way to start classifying bacteria. So how do we actually use this armor to tell different types of bacteria apart? The answer is a clever technique from over a century ago called the Gram stain. It was developed by a scientist named Hans Christian Gram. Here's the deal. Depending on how their cell envelope is built, some bacteria will soak up the purple stain, and we call those gram-positive. Others won't, and we call them gram-negative. It's one of the most fundamental ways we divide up the entire bacterial kingdom. All right, we've successfully breached the outer defenses. We are now inside the cell's main compartment, the cytoplasm. This is where everything happens. But unlike our own cells, there are no separate rooms or membrane-bound organelles. It's more like a bustling open-plan factory floor.
And the first thing you'd probably notice is what's not here. There's no distinct nucleus. Instead, all the genetic material, the cell's master blueprint, is bundled together in a region called the nucleoid. The DNA isn't protected by a membrane, it's just kind of coiled up right there in the cytoplasm, usually as one big circular chromosome.
But wait, that's not the only DNA in here. A lot of bacteria also have these other small circular pieces of DNA floating around, called plasmids.
I think the best way to imagine plasmids is like little swappable USB drives. They carry extra bits of code, genes, that aren't needed for everyday life, but can suddenly become superpowers. Think a gene for antibiotic resistance. And what's really cool is that bacteria can actually pass these plasmids to each other, like sharing a file. So if the DNA is the blueprint, what about the factories that build everything? Well, in prokaryotes, that job belongs to the ribosomes,
and they have a very specific size, 70S.
So what does that S mean? It stands for Swedberg unit, and it's not actually a measure of weight, it's a measure of how fast these tiny things settle during centrifugation. This 70S ribosome is built from two pieces, a big one called the 50S subunit and a smaller 30S subunit. Their one and only job is to read the genetic code and build every single protein the cell needs to survive. They are the protein factories.
Another really unique structure you'll find in here is the mesosome. This is basically just the plasma membrane folding in on itself, pushing into the cytosol. These folds create a ton of extra surface area, and they're involved in all sorts of important jobs, like helping to build the cell wall, copying the DNA, and even respiration.
Okay, our tour of the inside is done. Let's pop back outside and take a look at the tools these cells use to move around and interact with their environment, their external appendages. For any bacterium that's on the move, its main tool is the flagellum. This long, whip-like tail is made of 3 parts. The filament is the long part you see trailing behind. It's connected by a flexible piece called the hook to the basal body. And the basal body is the coolest part. It's a tiny rotary motor stuck in the cell wall that spins the filament around and propels the cell forward.
Now, besides flagella, you might see a couple of other things on the surface, but these are not for moving. First, you have fimbriae. These are small, bristle-like fibers all over the cell that act like Velcro, helping it stick to things, like rocks in a stream or even our own tissues. Then you have pili. They're a bit longer and are made of a special protein called pillin. They're also used for attachment.
Wow, okay, we have covered a lot of ground, right? From the outer armor to the inner factory to all the external gadgets. So let's put all the pieces together and look at the complete prokaryotic blueprint.
This chart really lays it all out beautifully. You've got the command center, the nucleoid, with those bonus plasmid USB drives. You've got the 70S ribosomes cranking out proteins. The whole operation is protected by the multi-layered cell envelope. And to get around and stick to things, the cell uses flagella for movement and fimbriae and pili for attachment. There's even storage units called inclusion bodies for nutrients. It's a masterclass in efficiency.
You know, we call these cells simple because they don't have all the fancy internal compartments that our own cells do. But that leaves us with a really fascinating question, doesn't it? How has this so-called simple design been so unbelievably successful that it's dominated just about every corner of our planet for over 3.5 billion years? It's pretty amazing to think about.